And we will now move on to our next speaker, who is Dr. Atsushi Sogabe, who obtained his PhD degree in 2007 from Ehime University in Japan. Now he works as an associate professor in Hirosaki University. Dr. Sogabe has contributed to the understanding of diversity of mating systems in syngnathids, and especially has been interested in the proximate and ultimate factors for maintenance of monogamy. Today, he will present one of his recent findings about microevolution in syngnathids, the loss of the vasotocin 1A1 receptor gene. So with that, take it away.
Thank you for your introduction. Just give me a minute. Okay, in this presentation, I'd like to talk about an interesting evolutionary event that has taken place in syngnathids. Through research over the past quarter century, it has become increasingly clear that neuropeptides and their receptors in the brain are deeply involved in the social behavior of animals. One of the most famous examples of this is that in birds. The distribution of vasoprecin receptors in the brain differs between monogamous and promiscuous species. In monogamous male prairie bird, vasoprecin 1A type receptors are distributed in higher density in ventral pallidum of forebrain, whereas in the promiscuous mountain bird, those are distributed in higher density in lateral septum. I was curious to know whether similar variations in the distribution of vasotocin 1A receptors according to the mating system also occur in syngnathids. So we wanted to compare the distribution of V1A receptors in the brains of 5 species of syngnathids with different mating systems. That is, Coesoictis haematoputerus, Hippocrates penicillus, and Hippocampus abdominalis as monogamous species, and Urocampus nanus and Syngnatha schurgeri as polygamous species. We decided to use in situ hybridization methods to examine the brain regions where the V1 receptor gene is expressed and start to develop primers and probes for in situ hybridization. Non-mammalian animal has arginine vasotocin, which is homologous to mammalian arginine vasoprecin. In mammal, only one type of V1 receptor is known, but in telos species, there are 2 types of V1A receptors, that is, V1A1 receptor and V1A2 receptor. So we need to clarify the distribution of both V1A1 and V1A2 receptors in the brains of syngnathids. Therefore, we created a cDNA library from RNA-seq of the brains of 5 species of syngnathids and searched for sequences similar to the V1A1 and V1A2 receptor genes of Japanese medaka for developing primers and probes for in situ hybridization. This figure shows a phylogenetic tree of the vasoprecin and vasotocin 1A receptor gene family based on the information available in the database. As you can see, vertebrates such as mammals, birds, and reptiles have only one type of V1A receptor gene. On the other hand, telos species have 2 types of V1A receptor genes, V1A1 and V1A2 receptor genes. In the RNA-seq data from 5 species of syngnathids, cDNA sequences with high homology to the V1A2 receptor genes of other fish were found, while sequences with high homology to the V1A1 receptor gene were not found. In other words, the V1A1 receptor gene was not expressed in the brains of syngnathids. Why was the V1A1 receptor gene not expressed in the brains of syngnathids? There are two possibilities. The first possibility is that the expression of the V1A1 gene is tissue-specific and is not expressed in the brain. Or, if it is expressed in the brain, it is only expressed at limited times depending on some condition. However, this is very unlikely. This is because in all fish species in which the expression of the V1A1 receptor gene has been examined so far, stably high expression of the gene has been found in the brain. And it is difficult to imagine that only syngnathids have no expression in the brain. Another possibility is that the V1A1 receptor gene has been lost from the genome of syngnathids. Therefore, the new objective of our research was to determine whether the V1A1 receptor gene exists in the genome of syngnathids. We tested this with two approaches. The first thing we did was to identify the arrangement of genes surrounding the V1A1 receptor gene on the genome map of the fish carrying V1A1 receptor gene. If the position and order of these genes are conserved across lineages, then we can look for these genes in the genome of syngnathids to see if there are V1A1 receptor genes around them or not. At the time we started the research, genome maps were known only for two species of syngnathids, Syngnathes aegas and Hippocampus comes, so we looked at them. These figures show the arrangement of the gene around the V1A2 and V1A1 receptor genes in Japanese medaka and 9-spined stickleback. As you can see, gene arrangement around the V1A2 and V1A1 receptor gene is basically well conserved. So we searched for these genes in the genome map of Syngnathes aegas and Hippocampus comes. The V1A2 receptor gene was found in a similar gene order in syngnathids. On the other hand, the same gene as those found in Japanese medaka and 9-spined stickleback were found in a similar arrangement in syngnathids. But the V1A1 receptor gene was not found in the vicinity. This result strongly suggests that the V1A1 receptor gene is lost in syngnathids. But it remains possible that the V1A1 receptor gene is translocated to a completely different position in the genome of syngnathids. As a next approach, we used 5 species of syngnathids whose genomes were known at the scaffold level to see if they had genes similar to the V1A1 and V1A2 receptor genes of medaka. The top 5 genes with the highest homology were selected and their respective genes were predicted. When this analysis was conducted for Hippocampus comes, the sequence with the highest homology to the medaka V1A2 receptor gene was included in the V1A2 receptor gene clade. However, the sequence with the highest homology to the medaka V1A1 receptor gene was found in the V1A2 receptor gene clade, indicating that Hippocampus comes carries the V1A2 receptor gene but not the V1A1 receptor gene. The same analysis was performed on the other 4 species. Also, sequences with high homology to the Medaka V1A2 receptor gene were found in all species. There were no sequences similar to the Medaka V1A1 receptor gene. Summarize the results. Also, the present study examined only a small fraction of the diverse lineage of syngnathids. It strongly suggests that the V1A1 receptor gene was completely lost in syngnathids. Why has the V1A1 receptor gene lost in syngnathids? At present, we do not know anything about the reason for this, but previous studies that examine the expression level of the V1A1 receptor gene in different tissues may provide a clue. It has been reported that the V1A1 receptor gene is expressed in various tissues other than the brain. But the common tissue with particularly high expression in various species is the male gonad. This suggests that the role of the V1A1 receptor gene, which is essential for male reproduction in other fishes, is not required in syngnathids. Fish that undergo external fertilization generally require large numbers of sperm to increase the fertilization success. In particular, fish that spawn in groups are exposed to strong sperm competition, so it is important to have certain characteristics of the sperm and ejaculation to increase the fertilization success. On the other hand, no evidence of sperm competition has been found so far in syngnathids. In the case of seahorse and some fishes, fertilization takes place in a closed space that is broad pouch, which allows for efficient fertilization with a small amount of sperm. The same can be said for other pipefishes and sea dragons that do not have closed brooding structure, as the brooding site and the genital pores are in close proximity to each other. Also, there is no evidence yet that the evolution of unique mode of reproduction in syngnathids, that is, male pregnancy, may have triggered the loss of the V1A1 receptor gene. In order to clarify this, we are currently investigating when the V1A1 receptor gene was lost in the lineage of Syngnathiformes. We are also planning to conduct experiments knocking out or knocking down the V1A1 receptor genes in model fishes to clarify the role of them in reproduction in males. That's all I have to say. Thank you for your attention.
Thank you very much for that excellent talk. We have a few minutes for questions. Does anyone have anything they'd like to ask? Feel free to go ahead and unmute yourself and ask your question.
Hi, Prof. Sogabe, very fantastic work. I
Thank you.
wondering, the function of V1A receptor, In Medaka, it has the gene. So any papers report that this gene involved in the sperm and the egg fertilization of this gene's function. in Medaka. If we look out, so maybe we can predict the phenotype of the knockout line. Yeah.
Yeah.
So-
Vasotocin peptides has many functions. It's depending on the receptor. So some studies suggest the social behavior. Vasotocin affects the social behavior of fish. Yeah. And
Okay. Thank you. Thank you. Very fantastic work.
Are there any other questions? If nobody else has a question, I'll ask a question. So you were talking about how the V1A1 receptor seemed to be in other fishes having specific expression in the testes. Does the V1A2 receptor have a similar sort of tissue specificity in other tissues? In either other fish or in syngnathids?
I don't know in expression in syngnathids, but in other fishes, V1A2 in other fishes, V1A2 receptor gene is expressed in many tissues, including brain, heart, and muscle.
So it doesn't seem to have that same importance in the reproductive tissues, perhaps.
Yeah.
Yeah. Interesting. I'll be excited to see what you find with your next steps in your research.
Thank you very much.
Were there any other questions for Atsushi?